back to MEDC by Lakshmi. So today we will be covering one of the important 5-mark question, which is the G protein coupled receptor. G protein coupled receptors. So by definition, Gpcr is G protein coupled receptor, or these are transmembrane receptors. It's present in the cell membrane itself, which control the cell function via 3 pathways: adenyl cyclase, mostly adenyl cyclase, and phospholipase C. Now what about the structure of this? So when we are seeing the GPCR, it will be like 7 helical structures, 7 alpha helical membrane. It will be hydrophobic, made up of hydrophobic amino acid. 3 will be in extracellularly present, 3 will be intracellularly present. Now this is coupled with something called the G protein. So G protein has got 3 subunits: alpha, beta, gamma, and we can also say it is heterotrimer. Now let's discuss the function of this GPCR. First I will teach you the easy way. So this is the whole process, and this thing I will teach you in the diagram which is from the textbook. I have also drawn it, but I think most apt will be learning it from the text itself, so I will be teaching you that. So first what will happen? Ligand binds to the receptor. The receptor will be there. Suppose adrenaline is coming, it will bind to the receptor. So when the binding is happening, it will give the signal to the G protein. I told the G protein is associated with this receptor. So once any ligand bind to the receptor, it gets activated, it gives signal to the G protein. So G protein has alpha, beta, gamma subunit. So activation through the receptor leads to the displacement of GDP by GTP in the alpha subunit. So once the signal reaches the G protein, what is happening? The alpha, beta, gamma. Alpha has the GTPase activity. Okay. So what will be happening? GDP is it's changed to GTP. Now GTP is bind with this alpha subunit. This as such will dissociate from the beta gamma subunit. Now this will undergo phosphorylation inside the cell. It's get terminated. Other details I will tell you in the diagram itself. Now let's see what are the types of G protein. Okay, alpha G protein, this part which is getting dissociated, it activates enzyme and it has GS GQ subunit. This G protein itself, alpha itself has GS and GQ subunit. Now let's discuss why we have these subunits and everything. Now types of G protein: GS, GQ, GI. Now suppose the one which is getting activated is GS alpha. If GS alpha is the one which is getting binded to the GTP, it will activate the adenyl cyclase. It is the effector pathway. It will activate this pathway. If this pathway is activated, ATP is converted to CAMP. It's increasing, and which will lead to the activation of pKa pathway. It's capital A, pKa pathway, and this will ultimately lead to the better excitation and contraction coupled reaction. Simple. Now what about GQ? GQ will activate phospholipase C pathway, which will activate the IP3 DAG. IP3 will increase the mobility of the calcium, and DAG will activate the PKC pathway. Now what about GI? I4 inhibition. It will inhibit the adenyl cyclase pathway, therefore decrease the CAMP. It will open the calcium channel and it will lead to hyperpolarization. Learn it by heart. It's simple also. Adenyl cyclase, increase CAMP, pKa pathway. GQ, phospholipase converted to IP3 DAG, IP3 mobilization of the calcium, DAG, PKC pathway activation. GI, the opposite of GS, the inhibit adenyl cyclase, decreases CAMP, opens potassium channel, leading to hyperpolarization. Now let's learn few of the examples of GPCR. I have written 4 examples. One will be the muscarinic receptors. Second will be the 5-HT1, I think serotonin 1 to serotonin 7, except for 5-HT3, alpha beta receptors, angiotensin receptor. And this is the diagram I drew. To understand it better, I'll explain. After this, I will explain it from the textbook. It is the same diagram as that of the textbook, but I have made it less complicated, I think. It's the same diagram. So this is an adenyl cyclase effector pathway. This is the adrenaline. This is the ligand. Ligand binds with the 7 alpha helical structure. This is the receptor coupled with this G protein, has 3 subunit alpha, beta, gamma, GS alpha. Once it binds with helical structure, this gives signal to this G protein. So G protein alpha GDP is converted to GTP. Now this GTP binds with this GS alpha, which will lead to the dissociation of it from the beta gamma subunit. Now there is GS alpha binded with the GTP. This unit will activate the adenyl cyclase pathway. In the adenyl cyclase, getting the signal, more ATP is converted to CAMP. It will activate the pKa pathway. So pKa pathway is getting phosphorylated and it is activating proteins such as troponin. So what will happen? It will lead to better excitation and contraction coupling will happen, and ultimately it will lead to increased cardiac contractility. This will be the drug effect. Okay, increased cardiac contractility. This is one of the example. In textbook there is even more examples. So this is how you should interpret that image. And yeah, once this pKa pathway is open, more calcium is coming from more calcium channel, more calcium influx will be there, and from intracytoplasmic also there will be calcium influx. That's it. Thank you. And the textbook video I will make a separate video and put in my YouTube channel, since time is more in this video. Thank you.